Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological character to classify plants into trees, shrubs, and herbs. He also divided animals into two groups: those which had red blood, and those did not. In Linnaeus' time, a two-kingdom classification system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergone several changes over the time. Though plant and animal kingdom have been a constant under all different systems, the understanding of what groups organisms be included under these kingdom have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Table 2.1 Characteristics of the five kingdom. Character: cell type: prokaryotic in Monera, eukaryotic in Protista, eukaryotic in fungi, eukaryotic Plantae, and eukaryotic Animalia. Cell wall: non-cellulosic polysaccharide plus amino acids in Monera, cell wall present in Protista, present with chitin in fungi, and present cellulose in Plantae and absent in Animalia. Nuclear membrane: in Monera absent, but in Protista, fungi, Plantae, and Animalia it was present. Body organization: unicellular in Monera and Protista, and multicellular and loose tissue in fungi, and tissue plus organ in Plantae, and tissue, organ, and organ system present in Animalia. Mode of nutrition: autotrophic, chemosynthetic, and photosynthetic, and heterotrophic, saprophytic plus parasitic in Monera, autotrophic, photosynthetic, and heterotrophic in Protista, heterotrophic, saprophytic plus parasitic in fungi, autotrophic and photosynthetic in Plantae, heterotrophic, holozoic plus saprophytic in Animalia. Table completed. R.H. Whitaker 1969 proposed a five kingdom classification. The kingdom defined by him were named Monera, Protista, fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 that was given gives a comparative account of different characteristics of the five kingdoms. The three domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryote kingdoms in the third domain, and thereby a six kingdom classification. You will learn about this system in detail at higher classes. Let us look at these five kingdom classification to understand the issues and consideration that influence the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, and the angiosperm under plants. The character that unified the whole kingdom was that all the organisms include had a cell wall in their cells. These placed together group which widely differentiated in other characteristics. It brought together the prokaryotic bacteria and the blue-green alga cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say, for example, Calaminodomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed the characteristic differences in their walls composition. The fungi had a chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryote organisms were placed in the kingdom Protista. Kingdom Protista has brought together Calaminodomonas, chlorella, earlier placed in algae within plants and both having cell wall, with Paramecium and Amoeba, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdom. This happened because the criteria for classification changed. These kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationship over time. An attempt has been made to evolve a classification system which reflect not only the morphological, physiological and reproductive similarity but is also phylogenetic, that is based on evolutionary relationship. In this chapter we will study characteristics of kingdom Monera Protista fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, we will be dealt separately in chapter 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow and deep ocean, where very few other life form can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped into 4 categories based on their shape: spherical, coccus, plural, that's plural is cocci; the rod-shaped bacillus, plural bacilli; the comma-shaped vibrium, plural vibrio; and the spirillum, plural is spirilla. Figure 2.1. Bacteria of different shapes. Though the bacterial structure is very simple, they are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotroph, that is, they depend on other organisms or on dead organic matter for food. 2.11 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty area, halophiles, hot spring, thermoacidophiles and marshy area, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure and this feature is responsible for their survival in extreme conditions. Methanogens are also present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and if motile flagellum. The cyanobacteria are also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous freshwater, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example: nostoc and anibina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume. Figure 2.2, a filamentous blue-green alga, that is nostoc. In fixing nitrogen in legume, roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Chlorella, typhoid, tetanus, and citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission, given in figure 2.3. Sometimes under unfavorable conditions, they produce spores. They are reproduced by sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Figure 2.3, a dividing bacterium. 2.2, Kingdom Protista. All single-cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic Protista to one biologist may be a plant to one another. In this book, we include chrysophytes, dinoflagellates, euglenoids, and slime molds and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the Protista cell body contain a well-defined nucleus and other membrane-bound organelle. Some have a flagella or cilia. Protista reproduce asexually and sexually by process involving cell fusion and a zygote formation. Chrysophytes. 2.2.1 Chrysophytes. These group include diatoms and golden algae desmids. They are found in freshwater as well as marine environment. They are microscopic and float passively in water currents. Plankton. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping cells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus diatoms have left behind large amount of cell wall deposit in their habitat. These accumulation over billions of years is referred to as diatomeous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producer in the ocean. 2.22 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cell. The cell wall has stiff cellulose plates on the outer surface. Most of them have a two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Wall plates very often red dinoflagellates. Example: Gonicolyx undergo such rapid multiplication that they make the sea appear red. Red toxins released by such large number may even kill other marine animals such as fishes. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have a two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other Smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Example, euglena. Slime molds. Slime molds are saprophytic protista. The body moves along decaying twigs and leaves, engulfing organic material under suitable condition. They form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable condition, the plasmodium differentiate and forms fruiting bodies bearing spores at their tips. The spores possess true wall. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air currents. 2.25 Protozoans. All protozoans are heterotrophs and live as a predator or parasites. They are believed to be primitive relative of animal. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have a silica cells on their surface. Some of them, such as entamoeba, are parasite flagellated protozoans. The members of these groups are either free living or parasitic. They have flagella. The parasitic forms cause death, cause disease such as sleeping sickness. Example, trypanosoma, ciliated protozoa. They are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity, gullet, that opens to the outside of the cell. The coordinated movement of rows and cilia causes the water laden with food to be stirred into the gullet. Example, paramecium. Sporozoans. These include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite which causes malaria, a disease which has a staggering effect on human population. Para 2.3 Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi. You must have seen fungi on a moist bed and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to parasitic fungus. Some unicellular fungi, example, yeast, are used to make bread and beer. Other fungi cause disease in plants and animals. Weight rust causing puccinia is an important example. Some are the source of antibiotics. In example, penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. Their body consists of long slender thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called syncytic hyphae. Others have septa or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as a symbionts in association with algae as lichens and with roots of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fishing, and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Fusion of two nuclei called karyogamy. karyogamy. Third, meiosis in zygote resulting in haploid spores. When a fungus reproduce sexually, two haploid hyphae of compatible mating types comes together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell 2, and however in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage n plus n, that is 2 nuclei per cell, occur. Such a condition is called dikaryon, and the phase is called dikaryon phase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which division occur, leading to the formation of haploid spores. The morphology of the mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. 2.31 Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and cynocytic. Sexual reproduction takes place by zoospore motile or by aplanospores, that is non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, and isogamous or ogamous. Some common example are mucor, figure 2.5a, rhizopus, the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. 2.32 Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, that is penicillium or rarely unicellular. Example, yeast Saccharomyces. They are saprophytic, decomposer, parasitic or coprophilous, growing on dung. Mycelium is branched and is septate. The asexual spores are conidia, produced exogenously on the special mycelium called conidiophores. Conidio on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are aspergillus, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicious. Basidiomycetes, commonly known forms of Basidiomycetes are mushroom fungi or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites. Example, rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus mushroom, figure 2.5c, Eustelgo smut, and Puccinia rust fungus. 2.34 Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phase of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into the classes they rightly belong to. It is possible that the asexual and vegetative stage have been one name and placed under Deuteromycetes, and the sexual stage another and placed under another class later. When the linkage were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stage of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is separate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria coletrichum and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are example of insectivorous plant, and Cuscuta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae include algae, bryophytes, pteridophytes, gymnosperm and angiosperm. Life cycle of plants has two distinct phases: the diploid sporophytic and the haploid gametophytic. They that alternate with each other. The length of the haploid and diploid phases and whether these phases are free living or dependent on other vary among different groups in plants. These phenomena is called alternation of generation. You will study further detail of this kingdom in chapter 3. 2.5 Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular, and their cells lack cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent feature of various phyla are described in chapter 4. 2.6 Viruses, viroids and viruses, viroids, plants and lichens. In the five kingdom classification of Whitaker, there is no mention of lichens and some acellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know what effect viruses can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not conditioned. Viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you recall viruses living or non-living? The name virus, that means venom or poisonous fluid, was given by Dmitry Ivanovsky in 1892. Recognized certain microbes as causal organism of the mosaic disease of tobacco. Figure 2.6a. These were found to be smaller than bacteria because they passed through bacteria-proof filter. M.W. Bazarnicki in 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants, and called the fluid as Contagium vivium fluidum, infectious living fluid. W. M. Stanley in 1935 showed that viruses could be crystallized, and crystal consist largely of protein. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No viruses contain both RNA and DNA. A virus is nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single stranded RNA, and viruses that infect animals have either single or double stranded RNA or double stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double stranded DNA viruses. Figure 2.6b, the protein coat called capsid, made of a small subunit called capsomere, protects the nucleic acid. These capsomere are arranged in helical or polyhedral geometric forms. Viruses cause disease like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic information, leaf rolling and curling, yellowing and vein clearing, dwarfing, and stunted growth. Viruses. In 1971, Teodina discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses, and hence the name viroid. The RNA of the viroid was of low molecular weight prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similarly incised to virus. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, BSE, commonly called as mad cow disease in cattle, and its analogous variants Creutzfeldt-Jakob disease, CJD in humans. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is called phycobiont, and fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas. Summary: biological classification of plants was proposed by Aristotle on the basis of simple morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationship. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. But bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoa. Protists have defined nucleus and other membrane-bound organelle. They reproduce both asexually and sexually. Members of kingdom Fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are the four classes under this classification. The Plantae include all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pteridophytes, gymnosperm, and angiosperm are included in this group. The life cycle of plants exhibit alternation of generation, gametophytic and sporophytic generation. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of this organism is holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like viruses and virids as well as the lichens are not included in the five kingdom system of classification. Thank you friends for listening this audio, and please post your comment if you want any kind of improvement. So friend, मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए बाय, and just stay tuned with biology critic and enjoy the knowledge.